Good afternoon, children. Last time we had started with our new chapter, that was sexual reproduction in flowering plants, where we had discussed various details about flower, the technical terms, then about the male gametophyte, their structure, development, and some additional terms about the structure of pollen grain, the sporoderm, the pollen kit, etc. Today we're going to start with our last topic in the male gametophyte, because after this we are starting with the female gametophyte. This new topic is development of male gametophyte. First and foremost, what is male gametophyte, children? It is just the other term for pollen grain. The development of pollen grain is called as precocious germination. Why? Because when the pollen grains are still inside the pollen sac in the anther lobe, they start germinating, right? That is, usually the germination should occur rightly after they are released. But because in most of the plants it happens inside the pollen sac, while it is still inside before pollination, it begins with the development, therefore precocious germination. Let us see stepwise. It is very simple, children. There are 5 steps to discuss. I'll send you the pictures of these so you can make up into your notes. The 5 steps are like this, broadly divided into 2 parts. Development of male gametophyte in angiosperms have been divided into 2 parts. Pre-pollination development and post-pollination development. This is the whole summary of development of male gametophyte. How it is going to occur? In stages A, B, C, D and E, which will end up the pre-pollination development. Let us see what all things are going to happen over here. Look at this diagram A. It depicts a pollen grain with the exine outer part, intine the inner layer. Then there is cytoplasm. The number of changes that are going to occur is going to start with the nucleus. Number 1. Nucleus grows in size and becomes bigger, right? 2. It shifts somewhere near the wall and it undergoes mitosis, right? The equational division, and now the nucleus will be divided into 2 cells. The cell that is a pollen grain now is divided into 2 cells. The tube or vegetative cell, which is larger, you can see that, this bigger part. And why it is bigger? Because it has abundant food reserve and has a large nucleus. Whereas the smaller cell formed by mitosis is called as generative cell, which is small and spindle shaped. It is very interesting, children, to tell you here only that one cell is called as a tube or vegetative. Why tube? Because it will be at the tip of pollen tube to be formed later. Whereas generative cell further by mitosis will divide into or generate 2 male gametes. Alright. Moving further, let us see the further changes in generative cell is that it is surrounded, or a layer of callose wall is laid down around the generative cell, which later on of course dissolves, releasing the generative cell. Alright. Then sometimes most of the pollen grains, they are released at 2 cell stage, that is tube or the vegetative cell and the generative cell. But some of them, they will be released at 3 cell stage later on. That will be going to form later. Let us see how. This generative cell or the nucleus now further divides by mitosis, giving rise to 2 male gametes. Let us see the number of vacuoles laid down in the meantime, right? And now after mitosis, the generative cell has generated 2 male gametes. In green color you can see, and the tube nucleus, as I told you, like a torch bearer, is already at the tip of pollen tube. The male gametes, they are surrounded by cytoplasmic sheath, and you can see callose plugs already there into the pollen tube that has emerged from the germ pore. What was germ pore? Into the diagram, the portion where exine was missing. So I hope you've understood. Once again, to end up pre-pollination development, the nucleus enlarged, moved to one of the sides by mitosis, divided into 2 cells, tube or vegetative cell and the generative. This was bigger with abundant food, etc., whereas this is smaller and spindle shaped. Layer of callose was laid down, which later on dissolves, setting the generative cell free. With further mitosis, the generative cell divided into two male gametes. I hope this is clear. Now, the second part of development of the male gametophyte is post-pollination. That means after pollination. Let us assume by various agencies of wind, water, etc., the pollen grains have landed on the stigma or the female gametophyte. In this case, remember children, the stigma usually is sticky, right? This stickiness is because of the fact that it is secreting some stigmatic secretions, all right? And a lot of water molecules the pollen grains will absorb from the stigma head. What will it absorb? Nutrients and water, and this absorption will be held by the germ pores. As a result, the tube or vegetative cell, that is the bigger cell, right? This bigger cell, this will enlarge to form pollen tube, and this is happening after the absorption through the style. Finally, the generative cell has already divided into two male gametes. So this is what I told you children. Some of the pollen grains, they are released at 2 cell and some are released at 3 cell with this layout 1, 2 and 3. We are counting all of them individual. Whereas here if we count, they were only 2. That completes the male gametophyte and its development. To move further, we are going to introduce with the female reproductive organs or the terms. Let us see first with the female gametophyte. Number 1, ovule or megasporangium. Ovule, we all know the structure, that is stigma, style and ovary, we all know they form pistil or a carpel. So whether we call pistil or carpel means the same thing, right? So ovule was a structure inside the ovary. This ovule will also be called as megasporangium. Now the second term is embryo sac or megaspore or megagametophyte or female gametophyte. There are 4 words children. Embryo sac, if you remember children, into this structure you've already studied earlier. This spherical to oval structure is embryo sac and we'll call it by 4 terms. Megaspore, megasporophyte, megagametophyte or female gametophyte. Then third term, pistil or carpel, we'll also call as megasporophil. Just contrast to microsporophil earlier, remember, now we are introducing megasporophil. Now monocarpellary, mono as we mean one. If the pistil consists of 1 carpel, will be called monocarpellary. Similarly, if it consists of 2 carpels, it will be called as bicarpellary. If it consists of 3 carpels, will be tricarpellary. And if more than them, there will be pistil consists of many carpels, will be multicarpellary. Sometimes there are two terms again. Sometimes pistil, the carpels are free, as seen as in the plants like Michelia and Ranunculus. Then we use the term epo. Epo means free, and carpels. So epo means free, right? Similarly, just opposite, if we say syncarpus, syn means fused or united, right? So syncarpus means when the carpels are fused. Example in poppy, scientifically called papaver, and hibiscus. So some 8 or 9 terms you must make a note and revise again and again. Now there's a general rule children. What it says is the number of locules. What were locules? Just to remind you, if you cut the TS of ovary, that is if you cut the ovary in this transverse section, then you see compartments like these, right? Into these compartments attached to the central axis are the ovules. So this is a locule. This is the ovule. And this is the ovary wall. So if you see a TS of an ovary, you will carefully just remind yourself or recollect how we had done the locule. So the rule says that the number of locules in a polycarpellary pistil is equal to the number of carpels. So what does it mean? If there are 5 locules, 1, 2, 3, 4, 5, that means looking at the TS we can say that the flower has 5 carpels. If it has something like 3 locules, 1, 2 and 3, with the ovules, right? Then it means that there are 3 carpels. So usually what I'm trying to tell you is number of locules is equal to the number of carpels, right? Then another term, unitegmic. Uni means one and tegmen means covering. Remember the integumentary system you've studied in humans, tegmen means the covering. Ovule with a single integument is called unitegmic, and this is seen in gamopetalous families. Gamo means the flowers which have fused petals, right? Then coming to bitegmic ovule, these are ovules with 2 integuments or 2 coverings, and this is seen in polypetalous. Poly means where the petals are fused, are free, sorry. Gamo means fused, poly means free. Please write these terms, children. Some of you really forget. Poly means free and gamo means fused, right? So if it says polypetalous, that means free petals. If it says gamopetalous, that means fused petals. Alright, and this is especially seen in monocot families. Coming to A means without. Ategmic means there is no tegmen or covering. Ovules without integument, santalum and loranthus, and these both are partial parasites as examples without any integument. Another new term, this is important, aril, sometimes also called as arm, ARM. What does it mean? This is apart from the 2 integuments, sometimes third integument is also present. In lychee plant, lychee, it is called as aril, and the same when present in asphodelus is called arm, right? And it is derived from funiculus. How would you understand just now, children? Let us see. This is the structure of a typical ovule. You can see in green color, there are 2 integuments. One the inner and one the outer. This is what we said. Bitegmic, sometimes one means unitegmic. Now when I say imagine there is a third integument to be formed. So from where it is going to form? From the funiculus. This structure, the base, small part which is attaching it, right, is called as funicle or funiculus. This is what we had discussed here, that in asphodelus, if it is the third integument, it is derived from funiculus. Another term called megasporogenesis. The process of formation of megaspore from MMC, that is megaspore mother cell, is called megasporogenesis. This we will start in detail next time, right? Now, this diagram is very, very important. You have to practice and learn how to label it and also describe from the diagram. What is this diagram of? It is an anatropous ovule or a typical ovule in transverse section. So this is the typical ovule. This is LS, sorry. Please check it. This is longitudinal section or anatropous ovule, of an anatropous ovule. Now, this is a typical ovule or a megasporangium. Let us see what all structures do we have to learn. Let us start from the base. First and foremost, we have a very small slender part, right? This is called as funicle or funiculus. The 2 parts are main of the ovule. This is one part. It is short and attaches it to the main placenta, right? The greater part of ovule is an oval body, right? And this is made up of a nutritive tissue called as nucellus. So if you divide the ovule, children, into 2 parts, we have funicle, this part of funiculus, and the oval swollen area called as the body, or made up of parenchymatous, that means the power of division, and oval structure consisting of nucellus. Nucellus is a nutritive part, enough of food reserves or the material for growth. So 2 parts I told you, funicle and the body or the nucellus. Now further, if we move up, you can see this ovule is actually an inverted part, right? This is how you have to imagine something as inverted like this, and it is called anatropous ovule. What are the features of anatropous ovule? It's a very most common type, very commonly seen. Body is inverted, that means this whole thing has kind of inverted. Micropyle is close to hilum. Now what is micropyle, children? Micropyle is a small gap between the 2 integuments, outer and the inner. So this is near the hilum. What is hilum? It is the point of attachment of funicle to the body. that is this funicle, wherever it attaches, you can imagine if this is funicle, only this short stem, then wherever it is attached to this whole body is called as hilum. So hilum position is obviously here, nowhere else, because it's attached over here, right? Then moving further, there's a portion of area we call as chalaza, right? Due to which this is called as the chalazal pole and this opposite one was called as the micropylar pole. What is chalaza, children? It is a swollen basal part of nucellus from where integument arises. That is the area, you can see from here only the integument is arising, right? Whether the first or the second, right? So this part where the integuments are arising is called as chalaza, right? Then the outer and inner integuments. Now coming to the most important part of this ovule is the female gametophyte, or there are many terms, children. This whole rounded oval structure called as embryo sac or megaspore or